Recording in progress.
Welcome everyone to this RRR series of the StudyMEDIC family. And today, for the brief 20-30 minutes, we shall discuss on the topic carbohydrate metabolism from biochemistry. And I am Dr. Maitreyee. I'll be leading you through this brief session. And the agenda, how it goes on the carbohydrate metabolism, on the glycolysis, TCA cycle, gluconeogenesis, HMP pathway, and the glycogen metabolism, where the lysis and synthesis will be seen in detail. Before we begin into all these metabolic pathways, we will take a look of the points that what are the different carbohydrates which we consume. So the carbohydrates we can classify as the monosaccharides, or they can be classified as the disaccharides, or they can be classified as the polysaccharide. Now this hexose, which is the monosaccharide, that is the glucose, examples being fructose, galactose, and the mannose. Now glucose, we all know it is the important one which is taking care of our sugar levels, and then the fructose is obtained via the hydrolysis, which is obtained from the sources like fruit juice, honey, hydrolysis of the cane or the beet sugar, or there is a polysaccharide called as inulin. And if this fructose is not properly digested because of the enzyme deficiencies, so the conditions called as hereditary fructose intolerance, where there will be fructose accumulation and leading to hypoglycemia. So this can be seen in. About the galactose, it is the source obtained from the hydrolysis of lactose. Lactose is a disaccharide which is obtained from the milk sugar obtained in the milk. Now, hereditary galactosemia is a clinical condition where the enzyme required for galactose metabolism is defective, that is, which is causing the condition like a cataract or mental retardation of such clinical conditions. Now we'll see about the pentose sugars. Pentose is another sector of monosaccharides. Pentose means they are having 5 carbon units, whereas hexose are having 6 carbon units. Now examples for pentose are the D-ribose, D-ribulose, arabinose, xylose, xylulose. So ribose, what is the source for this D-ribose? That is an important product received from the nucleic acid breakdown. And they are not only the nucleic acids like the DNA, RNA, but also the coenzymes like the ATP, NAD, FAD. So it is an important component of these coenzymes as well. And about the ribulose, it is obtained from the pentose pathway, that is pentose phosphate pathway or HMP pathway. So these are the 2 important products to be remembered from the pentose. Now we'll see about the disaccharides. The examples for disaccharides: sucrose, which is obtained from the cane or the beet sugar or the table sugar, whatever we call. What is it made up of? It is made up of glucose and fructose. Lactose, lactose is obtained from the milk, and what is it made up of? It is made up of galactose and glucose. So the enzyme which is required for lactose digestion is lactase, and if this lactase is deficient, the person will develop what is called as lactose intolerance, presenting with diarrhea, flatulence of these exaggerated situations. About the maltose, it is made up of 2 glucose units, and this one is obtained from the hydrolysis of the starch or the amylase. There is another product called as isomaltose, where it is again made up of 2 glucose units, but here the bond you see between the 2 glucose, it is 1,4. Here it is 1,6 bond. Again, this is obtained from the hydrolysis of the starch. Lactulose, it is galactose and fructose subproducts made up of monosaccharides, galactose and fructose. It is obtained from the heated milk, and it is not hydrolyzed in our intestinal enzyme by any intestinal enzyme. Thus, it can act as a mild osmotic laxative. Trehalose, again, it is made up of 2 glucose subunits, but you observe again carefully, the bond between the 2 glucose subunits, monosaccharides, is 1,1. So maltose is 2 glucose, isomaltose is 2 glucose, trehalose is 2 glucose molecules, but you see the bond is alpha 1,4, alpha 1,6 and alpha 1,1 bond between the two monosaccharide units. Now, whenever we consume the food, it can be the polysaccharide, it can be the disaccharide, or it can be the monosaccharides. In our mouth, the polysaccharide digesting enzyme called as salivary amylase is present, which will break down the starch into dextrin, and further continuing down in the small intestine, the pancreatic amylase will join in to break down the dextrins to further smaller subunits, that is the disaccharide maltose. Now, this maltose can be acted by the maltase enzyme to give out two monosaccharide units, that is glucose. Sucrose at the intestinal brush border, the sucrase enzyme is present, so that will break down and it will give the two monosaccharide subunits, that is glucose and fructose. Lactose digesting enzyme is lactase, made up of galactose and glucose. So now all these monosaccharides at the intestinal brush border, they will be absorbed. So there is a transporter for the absorption of the glucose called as sGLUT1, which is present at the intestinal brush border cells. So it is a co-transporter which is transporting the sodium and glucose together. So it is a glucose transport because it is dependent on sodium, hence the name sodium dependent glucose transporter. Now this glucose and the sodium will be taken up, and the sodium, which is naturally an extracellular component, will be thrown out of the cell by the sodium potassium pump. So 3 sodium will go out and 2 potassium will come in with the help of the sodium potassium pump. Now the glucose which is there inside the enterocytes will be taken up to the capillaries through the transporter called as GLUT2 transporter. So that is at the intestinal level, there is a GLUT2 transporter, the serosal surface of the intestinal epithelial cells. Likewise, there are many GLUT transporters present in the various sites of our system. So the GLUT1 is present in the brain mainly, kidneys and RBCs. GLUT2 we just saw, it is also not only in the intestinal cells but also liver and pancreas. GLUT3 again, neurons, brain. GLUT4 is an important thing to be remembered. So it is the skeletal, cardiac and adipose tissue. Now this is the only transporter which is mediated by insulin. So it is an insulin mediated glucose uptake. GLUT5, it is present in the testes and sperm, which is required for fructose transport. Now, once the cells have taken up the respective share of the glucose, it will be further metabolized by the glycolysis. So what happens in the glycolysis, you see? So this is happening in the cytosolic side of the cells. In every cell it is happening. So there are 3 key enzymes, that is the hexokinase, phosphofructokinase and the pyruvate kinase. Now what is the action of the hexokinase? Glucose to glucose 6-phosphate. The phosphofructokinase action is to convert fructose 6-phosphate to fructose 1,6-bisphosphate. Now aldolase will split this hexose molecule to 3,3 carbon molecule, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Now this glyceraldehyde 3-phosphate will go through the series of reactions to finally yield the pyruvate. So phosphoenol pyruvate to pyruvate. The another key enzyme is pyruvate kinase. So now this is dependent on the ATP, that is ADP is getting converted to ATP. Now you see here the hexokinase and phosphofructokinase. So there is a requirement of ATP molecules. So 2 ATPs are utilized here. At the level of 1,3-bisphosphoglycerate to 3-phosphoglycerate kinase, there is another ATP molecule which is produced by the glycolysis, and pyruvate kinase is also yielding the ATP molecule. So 2 are utilized, 2 are produced from 1 glyceraldehyde 3-phosphate, and there is NADH. So 1 NADH is equal to 2.5 ATPs. So from glyceraldehyde 3-phosphate, the aldolase, when it splits this fructose to glyceraldehyde 3-phosphate and DHAP, so 1 glyceraldehyde 3-phosphate will give 1 NADH, 2 ATP molecule. Similarly, DHAP, dihydroxyacetone phosphate, will get converted to glyceraldehyde 3-phosphate. Then again that will give us 1 NADH and then 2 ATPs. So net there will be 7 ATP production under the aerobic conditions. If there is an anaerobic condition prevailing in the cell, what happens is the pyruvate will be converted to lactate. For the coenzyme required for this lactate dehydrogenase is this the NADH. Where is this NADH coming from? This is the source of NADH. So under anaerobic glycolysis, there will be only 2 ATPs which will be produced, whereas under aerobic glycolysis, there will be 7 ATPs which will be produced. And one more important thing, so the production of ATPs in the cell, so where the 1,3-bisphosphoglycerate kinase and pyruvate kinase, they are helping. So these are the 2 examples for substrate level phosphorylation. Now this glycolysis under a good regulation, they are activated by insulin, whereas inhibited by the glucagon or excess availability of the ATP. So they will be inhibiting this 3 key enzymes. The 3 key enzymes is hexokinase, phosphofructokinase, and pyruvate kinase. Now, once that pyruvate is converted to acetyl-CoA, pyruvate dehydrogenase is the one which is converting to acetyl-CoA. Now, within the mitochondria, so this pyruvate which has gone to convert to acetyl-CoA, that acetyl-CoA will combine with the oxaloacetate to form citrate. Now, a series of reactions will happen in the citric acid cycle. The important highlights being the production of NADH. So, NADH or the direct GTP production. So, we have already seen in the glycolysis there was a NADH production under aerobic conditions. So here we can see 3 NADH is produced and 1 FADH is produced. 1 FADH is equal to 1.5 ATP and 1 NADH is equal to 2.5 ATP. Now, how many ATPs, so the pyruvate, it is converting to acetyl-CoA. So in glycolysis from 1 carbohydrate, that is glucose, we got 2 pyruvate. So now this 2 pyruvate will form the 2 acetyl-CoA. So from 1 acetyl-CoA, we are getting 3 NADH and 1 FADH and 1 GTP. So we have to count in the 2 acetyl-CoA entering to the another cycle of TCA cycle. So altogether, we get 20 ATPs from the TCA cycle. So when a glucose molecule is subjected for glycolysis and TCA cycle, altogether it will give us 32 ATPs. So the significance of this TCA cycle, it is the final pathway where the oxidation of the carbohydrate lipid protein happen. If there is a good amount of energy production, and it is also referred to as the amphibolic process because there is both catabolism and anabolism is happening. Anabolism means production of the new compounds like gluconeogenesis, fatty acid synthesis, porphyrin, synthesis of various amino acids, nucleotides. So these are all synthesized from the intermediates of the TCA cycle. So TCA cycle is not only the catabolic thing, but also it is the intermediates are able to produce us all these things. So hence it is referred to as amphibolic process. So you can see in this overview where the citrate is able to produce fatty acids, alpha ketoglutarate is able to produce the neurotransmitter GABA, and then it is able to produce purines. Succinyl-CoA is able to produce the heme, porphyrins. Aspartate is able to produce as the pyrimidines, purines, and other amino acids. So that's about the TCA cycle. So we finished with the glycolysis and then the TCA cycle. Let us take a look into the gluconeogenesis. The name it's telling, there is a production of new glucose molecule from a non-carbohydrate precursor, which can be, that is lactate, glycerol, glucogenic amino acids, or propionyl-CoA. Or it can be the intermediates of TCA cycle. So where exactly is this gluconeogenesis happening? In the liver, partly mitochondrial and partly cytoplasmic. So you have to remember this point. Glycolysis was happening in cytosol, TCA cycle in mitochondria. Now gluconeogenesis, it is partly mitochondria and partly cytoplasmic. Now, you can see this very nice slide. So you can see the pink arrow marks and the blue arrow marks. So the pink arrow mark is all coming downward. The blue arrow mark is all moving upward. So this is a glucose to pyruvate formation. You have learned it as the glycolysis pathway. Now the reversal of the things is all about the gluconeogenesis. Now we had seen 3 key enzymes of the glycolysis. That is hexokinase, phosphofructokinase, and pyruvate kinase. Key enzymes means they are the irreversible steps. So wherever there are irreversible steps, so there are the counter enzymes of the gluconeogenesis. For pyruvate kinase, to convert pyruvate to phosphoenol pyruvate, we have pyruvate carboxylase and phosphoenol pyruvate carboxykinase. So these are the two enzymes which is helping to produce the pyruvate to phosphoenol pyruvate. Now this phosphoenol pyruvate will go back all the way to form fructose 1,6-bisphosphate. Now fructose 1,6-bisphosphatase is the enzyme which will convert to fructose 6-phosphate. Now fructose 6-phosphate will easily reverse to form glucose 6-phosphate. Now it will encounter the hexokinase key enzyme of the glycolysis. So there is an alternate enzyme, that is glucose 6-phosphatase. This is the key enzyme of the gluconeogenesis. So what it will do, it is able to give back the free glucose molecule. It will hydrolyze the glucose 6-phosphate and give us the free glucose molecule. So in this way, so from the pyruvate, we are able to get back the glucose molecule. So we just saw from pyruvate to lactate, that is the lactate dehydrogenase. So the lactate can go to the liver and it is pyruvate to glucose. We saw in the previous slide how pyruvate to glucose it is forming. So this is a substrate which is called as the Cori's cycle. So from lactate, we are able to get back the glucose. Why lactate is produced in skeletal muscle? Whenever the muscle is going through the vigorous exercise, anaerobic condition sets in, pyruvate will be converted to lactate. That lactate will travel all the way to liver and help us to yield glucose molecule. So this is one of the metabolic pathway where the glucose is produced, that is gluconeogenesis is happening. This is referred to as Cori's cycle. Then we can see another example how amino acid is able to give us the formation of glucose. So alanine, which is the structural component of the skeletal muscles. So this alanine will be derived from the skeletal muscle. Alanine to pyruvate formation, there is a ALT enzyme, alanine transaminase. That enzyme will convert to pyruvate. Now pyruvate to glucose, we have learnt in gluconeogenesis pathway. So this is example where it is called as glucose alanine cycle. This is a glucose alanine cycle. Also it is referred to as Cahill cycle. From the glycerol, glycerol is an abundant source which is available in the adipose tissue. This glycerol kinase and the glycerol 3-phosphate dehydrogenase will help us to produce the intermediate of glycolysis, that is dihydroxyacetone phosphate. Now once the intermediate of TCA cycle is formed, then it can easily go to the point of formation of glucose. So you can fructose 1,6-bisphosphatase and glucose 6-phosphatase. So the glycerol, that is the triacetyl glycerol, when it breaks down, it will give us to 3 fatty acids and the glycerol. So even glycerol, which is the breakdown of the triglycerides, is able to generate as the free glucose for us. Even propenyl-CoA, which is an odd chain fatty acid, it has got a 3 carbon in the structure. Even this odd chain fatty acid is able to produce us the glucose molecule. So how does it happen? Propenyl-CoA with the help of carbon dioxide and biotin will form methylmalonyl-CoA, and then methylmalonyl-CoA to form succinyl-CoA. Now succinyl-CoA, it is the intermediate of TCA cycle. So that will form oxaloacetate, oxaloacetate to pyruvate, pyruvate to phosphoenol pyruvate, and then the glucose. So that is the way how the glucose can form even from the odd chain fatty acid, that is the propenyl-CoA. Now, 2 important things to remember here, biotin and vitamin B12. So if B12 is not there available, then methylmalonic acid can be excreted in the urine. If biotin is not available, then this propenyl-CoA to d-methylmalonyl-CoA will not be happening. And biotin will always help us in the reactions of carboxylation reactions. Carbon dioxide is involved in. So the significance, liver is the only organ to replenish the glucose. So wherever the situations like hypoglycemia, so liver is the one to take care of our system. And also it clears the lactate and glycerol from the muscle and adipose tissue respectively. So when does gluconeogenesis happen? That is, the hormones like epinephrine, glucocorticoids, glucagon. So if these are the levels high, like when the person is starving, fasting, so under the circumstances, these hormones will be elevated. They are the one which will be inducing the four key enzymes of the gluconeogenesis. What is the action of insulin? Insulin has got a repressive role on these four key enzymes. So that's about the gluconeogenesis. Now we'll see about into the glycogenolysis. Lysis is breakdown. So glycogen, it's a polysaccharide which is stored in the skeletal muscle and in the liver. So which is made up of glucose molecule. Now glycogen is a highly branched structure. You can see the branching point is made up of the bond alpha 1,6. So now the key enzyme of the glycogen breakdown is the glycogen phosphorylase. What does it do? It will split off all these bonds between the glucose molecules, and the glucose 1-phosphate is freely released. Now this length is reduced. Now the transferase enzyme, the debranching enzyme, it will remove this segment and it will expose this bond, that is alpha 1,6. Again, the debranching enzyme, that is alpha 1,6 glucosidase activity is there. So what it will do, it will release the free glucose molecule. So from here we had seen two branches connected with the branching point. Now you see, after the debranching enzyme activity, we are left with one single chain of the polysaccharide molecule. Now we want free glucose from glycogen. So what happens is, so the glycogen phosphorylase will further come into the action. It will again start chipping at these bonds, that is the 1,4 bonds. So the glucose 1-phosphates are again sequentially released. So this glucose 1-phosphate to glucose 6-phosphate will be converted by the enzyme mutase, and then the glucose 6-phosphatase enzyme will convert glucose 6-phosphate to glucose. So hence again we get from glycogenolysis, with the action of these two enzymes, mutase and the phosphatase, a free glucose molecule is obtained. So what is the key enzyme of this? Which is the key enzyme of the glycogen breakdown? Glycogen phosphorylase. Now what about the glycogen synthesis? Whenever the person has well-fed, body will try to build up the reservoir glycogen. So under the action of insulin, whatever the amount of glucose the person will consume, that will be deposited as the glycogen. So glucose it will not directly build as glycogen. It requires an activation. So that activation, how does it happen? Glucose 1-phosphate will be combining with the UTP to form the UDP glucose. Now this UDP glucose, with the help of glycogen synthase, which is the key enzyme of glycogen synthesis, and the branching enzyme, so what they will do, so they help in building up the glycogen molecule. As you can see in this slide, which is showing up from the bottom up. So glucose to glucose 6-phosphate, glucose 6-phosphate to glucose 1-phosphate. Now glucose 1-phosphate will combine with the UTP to form UDP glucose. Now there is a primer which is required called as glycogenin. Now this UDP glucose with the glycogenin, when they are acted by the glycogen synthase, the glucose molecules are added to the primer. So there is a continuous reactions will be happening, and wherever the branching points are required, the branching enzyme will help to establish the glycogen. So finally the two enzymes, that is glycogen synthase and branching enzyme, are required to build the glycogen molecule. So we finish with the glycogen metabolism, that is glycogenolysis, where the glycogen phosphorylase was the key enzyme, and glycogen synthesis, that is the glycogen synthase, was the key enzyme. Now we will take into the last pathway to see, that is HMP pathway, or it is called as HMP shunt pathway or hexose monophosphate pathway. Now an overview of this pathway: whenever a person is in a well-fed state, around 10 to 15% of the glucose is diverted for this shunt pathway. So majority will be going through glycolysis and then TCA cycle, but around 10 to 15% will be shunted to this HMP pathway. That's why it is the name shunt. Okay, so it has got two phases, oxidative phase and then the non-oxidative phase. So cut short in a very brief important points to say about HMP pathway. In oxidative phase, what happens is glucose 6-phosphate is converted to 6-phosphogluconate. So here the enzyme is glucose 6-phosphate dehydrogenase. There is a production of the NADPH coenzyme. Another NADPH molecule is produced in the same process. So we get ribulose 5-phosphate and then the ribose 5-phosphate. So the two important things to highlight over here is the NADPH formation to NADPH. Now, what is the role of this NADPH, this coenzyme? It is an important precursor synthesis for various, the products like fatty acids, the steroids. For all these things, it is required. And then there is another enzyme which is there in the required for the HMP pathway, that is the trans-ketolase enzyme, trans-aldolase enzyme. The trans-ketolase is dependent on the thymine pyrophosphate, which is the coenzyme of vitamin B1. So it is required for conversion of the ribulose to glucose 6-phosphate. So these are the two important enzymes of the HMP pathway, the glucose 6-phosphate dehydrogenase, and then the ketolase, and then the aldolase. So continuing with this ribose 5-phosphate, what does it help? Where does it help? Nucleotide formation, coenzyme formation, the various the structural nucleic acids, and then the RNA formation. So what are the important products of HMP pathway? One is NADPH and then the ribose 5-phosphate. So we learned about NADPH, why it is required. It is required for the fatty acids and the steroid formation. Ribose 5-phosphate, it is required for the nucleic acids requirement. Now this glucose 6-phosphate dehydrogenase, if this enzyme is deficient, it can lead to hemolysis. Okay, so why there is a hemolysis when the person is having glucose 6-phosphate dehydrogenase, and these subjects, if they consume the medications which can cause the free radical, burst of free radicals, so it can cause hemolysis. How does it happen? So free radicals, one of the example is hydrogen peroxide. So this can disrupt the red blood cells and it can cause the hemolysis. How does this HMP pathway will help is this NADPH. So this NADPH, as you can see here, so it is helping to convert glutathione to reduced glutathione. So the enzyme is glutathione reductase. So this, the source of the hydrogen is coming from NADPH. So the glutathione, oxidized glutathione becomes reduced glutathione. Now this reduced glutathione will convert hydrogen peroxide to a non-toxic water molecule. So the key enzyme here is the selenium, which is a cofactor for glutathione peroxidase. Glutathione peroxidase will catalyze this vital reaction. So if NADPH is, say, if it is not available, or if this, the selenium is not available, or glutathione peroxidase, for some reasons these enzymes are defective, then the levels of this hydrogen peroxide can go high and it can cause the hemolysis. So why NADPH can there be deficient? Because if this glucose 6-phosphate dehydrogenase is not available, congenital deficiency of this enzyme, so the NADPH is not produced, and then there can be a toxic levels of free radical accumulation causing hemolysis leading to jaundice. So the importance of this HMP pathway, so one is DNA RNA formation. It is required for NADPH formation. This NADPH in turn is required for fatty acid synthesis, cholesterol, steroid synthesis. We just saw how it is required for free radical scavenging, and we learned that glutathione peroxidase, which is a selenium dependent, was the important enzyme over there, and it is required to maintain the RBC membrane integrity. And this NADPH also helps in detoxification functions of the various drugs. And whenever the hemoglobin is exposed for some vehicular exhaust, the hemoglobin, the ferrous form can convert it to ferric form. It becomes methemoglobin, and the oxygen carrying capacity, it will not help in oxygenation. We want to revert it back. So NADPH will help in reverting back to the methemoglobin to hemoglobin formation. So thus NADPH has a vital role in oxygenation as well through conversion of methemoglobin to hemoglobin. It prevents the formation of methemoglobin basically. And never to forget this, the source of NADPH is because of the important enzyme glucose 6-phosphate dehydrogenase, and deficiency of this can lead to hemolytic anemia. So wherever the person's, one of the classic examples, antimalarial drugs. So these drugs can impose, increase in the bilirubin levels because of the lysis of the red blood cells in patients who are having G6PD deficiency. So to summarize, in this series, in this today's episode, we learned about the carbohydrate metabolism, where we saw after the digestion of the carbohydrate, so it will be first subjected for glycolysis. After that, it will go for the final oxidation, where the good amount of ATPs are produced in the TCS cycle. And whenever a person is having fasting or person is having low glucose levels, the liver is the one which is taking care to produce the glucose, that is via gluconeogenesis pathway. And we saw the various substrates, that is Cahill cycle, Corey's cycle, glucosylanin cycle, all these things we saw how even the glycerol was helping. And in a well-fed state, there is a pathway called as shunt pathway, that is HMP shunt pathway, which is activated, which was helping us to produce NADPH and the ribose 5-phosphate, and glycogenolysis, that is the breakdown of the glycogen, which is there in the liver and also in the skeletal muscles. So in fasting or the starvation, the glycogenolysis will happen. So till what time the glycogen is able to take care of the glucose supply? It is around 48 hours. So till 48 hours, the glycogen is able to supply the glucose molecule. Finally, in well-fed state, the glycogen synthesis. So these are the major metabolic pathways: glycolysis, TCA, gluconeogenesis, HMP pathway, glycogenolysis, and glycogen synthesis. So these are the various pathways of carbohydrate metabolism required for the exams. I hope this video will help you in coming up exams to prepare well. So do follow us. Click on the follow subscription link and follow the next series to come up. So all the best. Thank you very much.
Recording stopped.
Bisma? Sukanya?
Yes, ma'am.
How was it? Feedback?
Good, ma'am.
Okay, thank you. So I have taken care of the time as well, because I saw in morning Dr. Soumya was mentioning to take care of the time as well. So I hope to I managed to cover in 34 minutes. So shall I log out, Bisma and Sukanya?
Yes, ma'am.
Thank you, dear. Thank you so much for your back-end support. Sukanya, thank you for joining in.